We'll be discussing gel electrophoresis and PCR. PCR, commonly known as the polymerase chain reaction, is an extremely useful biological technique that allows one to run an analysis on DNA that is either too small or too degraded to be characterized. This is due to it being able to exponentially amplify DNA in only a few hours. Thus, it is possible to characterize small portions of discovered DNA, such as those found in a crime scene or extinct dinosaurs. Initially, the process was extremely tedious, as the polymerase was denatured and had to be replenished in each step. In addition, it required 3 water baths set at different temperatures and a technician to consistently keep moving the reaction vessel from bath to bath and replenishing the DNA polymerase. Nowadays, the process has become much more simple, primarily due to the use of the heat-stable DNA polymerase of Thermus aquaticus, a thermophilic bacteria. This is also known as Taq DNA polymerase, and it is not inactivated by the high heat required in the PCR reaction. Thus, it only needs to be added once. Finally, the first automated PCR machine allowed cycling of temperature and fluids, further simplifying things. And now we will cover the requirements and process of the polymerase chain reaction.
For successful PCR reaction, there are 5 crucial ingredients. First, you need a template DNA. You're going to need large quantities of the 4 nucleotides, large quantities of the primer, DNA Taq polymerase, and a buffer. There are 3 main steps to the PCR reaction. The first step is denaturation, in which the DNA must be heated to 94 degrees Celsius so that the DNA can actually unwind and create 2 single strands. The second step is the annealing of the primers. The Taq polymerase needs to build a new strand of DNA. To do this, it must attach to the template by sticking to the DNA. In order for it to stick to the DNA must be cooled to 55 degrees Celsius for 30 to 60 seconds. This allows for optimum adherence of the primer itself. The primer allows the Taq polymerase to know where to begin the replication. The third and final step to PCR is extension, in which the Taq polymerase incorporates complementary nucleotides to the DNA itself. The polymerase works best at 72 to 75 degrees Celsius, at which the DNA nucleotides will actually be added to the strands. When we repeat this process over and over again, the new strands themselves will be used as templates so that the number begins to grow exponentially. Now on to gel electrophoresis.
Agarose is derived from a modified molecule found in seaweed. This creates a porous gel through which DNA fragments can travel. The purpose of gel electrophoresis is to sort strands of DNA based on their size. The larger strands will stay closer to the well near the negative pole, while the smaller fragments travel farther down the gel towards the positive end.
For example, if you think of the larger fragments as the elephant and the smaller fragments as the bunny, you can see that the agarose is this entire forest over here. The larger fragments won't be able to make it through the forest as well as the bunny. So, technically the elephant could knock down all of the trees, but for example, he can't and is therefore hindered, while the bunny can make it all the way to the other side.
On the negative side of the gel, there are wells where the DNA samples are loaded. Since DNA is negatively charged, it will be pulled towards the opposite and positive side of the gel.
Lithium bromide has already been added to the gel by the lab coordinator because it's extremely carcinogenic and no one wants to really touch that. So it's added so that we can see the DNA using a UV light when it's put under a ultraviolet transilluminator.
Gel electrophoresis can be used for criminal investigations. If DNA from a crime scene is mapped and DNA from a suspect is mapped and compared, then the guilt of that suspect can either be denied or confirmed.
But in this experiment, we used it mostly to find out whether the mutation was a deletion or an addition in the C. elegans UNC22 gene, which you can see right here. It was clearly a deletion because the natural or wild type had more base pairs than did the UNC22 gene, so it traveled further and indicated that the number of base pairs are significantly decreased.